7. Cell cycle, AAQ Medical Science Unit 1. The cell cycle. The phases of the cell cycle. Interphase, G1 phase, cell growth, protein synthesis, increase in numbers of organelles and preparation for DNA replication. S phase, DNA replicates, and G2 phase, protein synthesis, rapid growth, checks for DNA damage, and preparation for nuclear division, development of the spindle. M phase or nuclear division, you've got mitosis, which is prophase, metaphase, anaphase, telophase. The products of mitosis are two diploid daughter cells genetically identical to each other and to the parent cell. Meiosis, two divisions, each with 4 stages and a short interphase in between. The products are 4 haploid daughter cells, gametes, each genetically unique, and that the diploid number of chromosomes is restored at fertilization. The significance of meiosis as a source of genetic variation. Comparisons of mitosis and meiosis. Locations, processes, functions, and products. Stem cells and differentiated cells. Division. Differentiated cells can no longer divide, but there are adult stem cells in all tissues which can divide. The role of adult stem cells in tissue repair and growth. When the cell cycle is disrupted, cancer as a result of inappropriate cell proliferation due to the cell failing to recognize signals for when to stop dividing and undergo apoptosis. The role of cytology screening in detection cancer. Cell death. Cells undergo a number of divisions, the Hayflick's limit, before undergoing apoptosis. The difference between apoptosis, programmed cell death, and cell death due to damage, injury, loss of blood supply, or infection, necrosis. Totipotent is stem cells with the potential to differentiate into totally any type of cell. Haploid, an organism or cell having only one complete set of chromosomes. Diploid, an organism or cell having 2 sets of chromosomes. Homologous chromosomes, a pair of chromosomes that are the same size, same appearance, and same genes. Somatic cells, body cells. Gametes, a haploid male or female germ cell that is able to unite with another of the opposite sex in sexual reproduction to form a zygote. Cell cycle, series of events that cells go through as they grow and divide. G1 phase, stage of interphase in which cells grow and perform its normal function. S phase, the synthesis phase of the cell cycle, the portion of interphase during which DNA is replicated. G2 phase, stage of interphase in which cell duplicates its organelles. Cell cycle checkpoints, mechanisms that monitor the preparedness of the eukaryotic cell to advance through the various cell cycle stages. Histochromatids, identical copies of a chromosome. Full sets of these are created during the S subphase of interphase. Centromere, area where the chromatids of a chromosome are attached. Bivalent, a pair of homologous chromosomes. Alleles, different forms of a gene. Mitosis definition occurs in body or somatic cells used for growth, tissue repair, and replacement of cells using stem cells. Reasons why it's important to produce genetically identical daughter cells for asexual reproduction for growth, repair, and replacement. Phases of mitosis: prophase, metaphase, anaphase, telophase. PMAT. Prophase, chromosomes condense and become visible. Nuclear envelope dissolves, spindle forms, centrioles form. Metaphase, chromosomes line up at the equator or middle of the cell. Each chromatid is attached to a centromere. Anaphase, chromatids are pulled to opposite poles of the cell. Telophase, the chromosomes uncoil and the nucleus reforms. Cytokinesis, division of the cytoplasm. Meiosis definition: a type of cell division that results in 4 haploid daughter cells, each with half the number of chromosomes haploid of the parent cell and which are not genetically identical, as in the production of gametes. Fertilization definition: fusion of male and female gametes to form a zygote where the full diploid number is restored. Where meiosis occurs: testes and ovaries. Why meiosis is necessary: to keep the chromosome number constant every generation. How variety is brought about in meiosis: crossing over and independent assortment, which occur in meiosis 1. Meiosis: 2 divisions, each with 4 stages and a short interphase in between. The products are 4 haploid daughter cells, gametes, each genetically unique, and that the diploid number of chromosomes is restored at fertilization. The significance of meiosis as a source of genetic variation. So, mitosis occurs in somatic cells, 1 division, PMAT, no pairing of homologous chromosomes, crossing over doesn't occur, it's for growth, repair, and asexual reproduction. It produces genetically identical cells, 2 daughter cells, diploid, same as the parent, identical to the parent and each other. Meiosis in ovaries and testes has 2 cell divisions, meiosis 1 and meiosis 2, so PMAT happens twice. Homologous chromosomes pair in prophase 1. Crossing over occurs in prophase 1, increasing genetic variation. Sexual reproduction, gamete formation, produces genetically different cells, 4 daughter cells, haploid, half the parent, genetically different from parent in each other. Stem cells. Division or differentiated cells can no longer divide, but there are adult stem cells in all tissues which can divide. Role in tissue repair, damage response. When tissue is injured, adult stem cells are activated to differentiate into the cells they need. Replacement of cells replace dead or damaged cells. Inflammation modulation. Some adult stem cells help regulate the immune response and reduce inflammation during healing. Role in tissue growth, homeostasis, maintain normal cell turnover in tissues and skin. In younger individuals, they contribute to growth and development of organs and tissues. Adaptation in response to increased demand, they help expand tissue capacity. Cancer as a result of inappropriate cell proliferation due to cell failing to recognize signals for when to stop dividing and undergo apoptosis. Role in cancer detection, early detection. Cytology can identify precancerous cells like a pap smear for cervical cancer. Screening, used in healthy individuals to detect early signs of cancer, used when symptoms of abnormal screening results suggest cancer. Monitoring, cytology can also help monitor treatment effectiveness and detect recurrence of cancer.